Welcome to the 6th annual CanVECTOR conference and our second virtual one. Create, connect, collaborate, promoting excellence in knowledge and practice to prevent venous thromboembolism. Next slide, please. My name is Cynthia Wu, and I will be one of your co-chairs today. I'm an associate professor in the Division of Hematology at the University of Alberta, Edmonton, Canada. I did my hematology and thrombosis training in McMaster University and spend the majority of my practice dedicated to thrombosis since returning to Edmonton. I have been a member of CanVECTOR since its inception and am very honored to be a co-host for this year's conference.
Delighted to be with you, Cynthia, today. Hello, everyone. And I'm Maha Othman. I'm a professor at the Department of Biomedical and Molecular Sciences at Queen's University in Kingston, Ontario. And I'm a clinical pathologist with specialization in hematology and subspecialization in laboratory hemostasis. I also trained in molecular genetics of hemophilia, von Willebrand disease, and platelet function defects. And my thrombosis research interests are focused on pregnancy, cancer, and hormonal contraception. I have also been an active member, but just for 2 years, so fairly new, and I'm honored and delighted to be co-chairing the conference this year with Dr. Wu. Next, please. So we are proud to present the conference this year in collaboration with Thrombosis Canada, and our CanVECTOR planning committee has reconvened earlier in the spring this year. And we put together the conference, and earlier we had wished for an in-person or a hybrid model, but obviously switched to the virtual model. And the planning committee for CanVECTOR has been, the diversity of it has been really crucial to shaping up the conference and choosing the diverse and relevant topic that fit with the CanVECTOR mission and also the theme for the conference this year. And I want to thank each member of the committee for putting this together. Starting with Dr. Eric Sang, who's the Thrombosis Canada representative and the past co-chair, our wonderful patient partner, Danielle Monroe, and our CanVECTOR trainee, Yan Zhu. And I must say that Yan has contributed substantially to the theme of the conference this year. Two wonderful research coordinators, those are Sophie Florencio and Michelle Zondak. And I want to say that none of this would have been possible without the generous support and countless hours put behind the scenes by two people. Those are Jesse Mikalok, who's the manager network operation, and Kaylin McGillivray, who's the manager for communication and digital media. So thank you all so much for putting the conference this year. Next, please. So for those of you who are new to CanVECTOR, this is a pan-Canadian patient-oriented community development program that is centered on venous thromboembolism. And when I say VTE, we actually target three main aspects: research, training, and knowledge translation. Our mission is to decrease the health, social, and economic impact of VTE, not only on individuals, but also on their families and Canadians as a whole. Next, please. So our membership has grown for over 222 since inception in 2015. Now, CanVECTOR, again, for those who are new, the way it was developed, this is nationally in collaboration with academic non-for-profit, but also industrial partners and stakeholders. We have two co-directors, those are Dr. Susan Kan and Dr. Mark Roger, who you will be hearing from later this morning. And our membership is very diverse. We actually have senior, mid-level, early career researchers who are interested in VTE, both on the clinical and the basic science sides, but we also have research coordinators, nurses, biostatisticians, pharmacists, and as well patient partners with experience and expertise in healthcare or health science research. I want to take this opportunity to encourage you to become a member and join our 222 so far. And this is simply by visiting the website and filling a simple form. Tell us who you are, what your research interest is, and explore the many opportunities on the website and how you can participate and support CanVECTOR. Next, please.
In line with our mission and living through together this COVID-19 pandemic, which challenges us on so many levels, we chose our theme this year to be create, connect, and collaborate, promoting excellence in knowledge and practice to prevent venous thromboembolism. Next slide, please. We have a very wonderful program for you today, and I'm super excited about it. All the times that are listed on this slide will be in Eastern Standard Time for everyone who, like me, is in a different time zone. To go over this quickly, our first keynote speaker today will kick off in just a few minutes, Professor Andreas Greinacher will be presenting on vaccine-induced immune thrombotic thrombocytopenia, or VITT. We'll then take a short break and reconvene at 11:15 a.m. to hear from Dr. Ilya Makedonov, who received the Thrombosis Canada CanVECTOR Fellowship Award in 2020. His presentation will be on anticoagulant selection for reducing the risk of post-thrombotic syndrome. And despite having an all-virtual conference this year, we were able to host the CanVECTOR Scientific Abstract Competition. We received 14 high-quality abstracts, and we have invited the authors to share their work in a virtual poster session. You can find these abstracts throughout the day on the poster tab of your conference platform, but the authors themselves will be available between 11:45 a.m. and 12:45 p.m. on the platform to chat about their work and answer any questions. Additionally, these abstracts will be published in a special edition of Research and Practice in Thrombosis and Hemostasis. At 12:45 p.m., we are very excited to welcome a team of patient partners and faculty from the Ottawa Hospital to present Patient Experience Storytellers, the Power of Stories in Healthcare. The workshop will explore the potential and relevance of patient stories in healthcare and research, and how these stories humanize our research. Next, at 2:30 p.m., CanVECTOR's own Patient Partner Council has developed an interactive workshop to explore how patient partners connect with researchers and contribute to projects across CanVECTOR. At 3:15 p.m., the top 3 rated abstracts in the CanVECTOR Scientific Abstract Competition will be presented. And last but not least, we will close the day with our second keynote presentation from Dr. Michelle Scholzberg, who will talk on her experience leading the RAPID trial, a recently published RCT comparing therapeutic dose anticoagulation to standard dose anticoagulation in COVID-19 patients admitted to the medical ward. Just a few housekeeping issues. We encourage you to maximize your conference experience by visiting exhibitors and posters during the break. And don't forget to take advantage of the networking tab on the conference platform to chat with your colleagues throughout the day. A quick reminder, there will be Q&A sessions included with every talk. Please just use the chat function on your screen to enter questions. And finally, we would love if you enhance the conference experience by tweeting about what you see and hear today using hashtag ThrombosisConference2021. Next slide, please.
So now we are delighted to welcome Professor Andreas Greinacher as our first keynote speaker for today. Professor Greinacher is specialized in transfusion medicine, immune hematology, and clinical immunology. He's a full professor and head of Department of Transfusion Medicine at the University of Medicine Greifswald in Germany. His research interests are congenital and immune-mediated thrombocytopenia, especially the heparin-induced thrombocytopenia, and particularly interested in the application of biophysics to understand the molecular mechanisms of antigenicity of endogenous proteins. He has published over 500 papers on these topics. And we have lived throughout the COVID-19 pandemic and the vaccination era, and we've witnessed all how VITT has become a very hot topic since last March. And less than a few months, there has been a substantial amount of publication on the diagnosis, on the treatment, on the pathology or potential pathology of this syndrome. Now, Professor Greinacher has been one of the very first few scientists who described patients with this syndrome, established together with many experts around the world on the laboratory test for an appropriate diagnosis of this syndrome, and also the proposed potential theory behind it. So I'm looking forward to hearing an update on VITT from Professor Greinacher. And without further ado, I would like to thank him again for being here with us today. And let's turn it over to him.
Yeah, Maha and Cynthia, thank you very much for the kind introduction. Ladies and gentlemen, dear colleagues and friends, thank you very much for the great honor to give the keynote lecture to your conference. I'll speak today on vaccine-induced immune thrombotic thrombocytopenia. These are my disclosures, which will not influence the content of my talk. Thrombocytopenia and thrombosis after vaccination are not infrequent. All COVID-19 vaccines are associated with an increased rate of thrombocytopenia causing ITP. All COVID-19 vaccines are associated with an increased rate of thrombosis like DVT or pulmonary embolism, and adenovector-based vaccines are associated with an increased risk also for combined thrombocytopenia and thrombosis called VITT or VITT. And this will be the focus of this talk. The structure of the talk is in 3 parts. I will address the known, the new, and at the end, briefly, some unknown aspects. All started with a clinical observation end of February 2021 that several unusual thrombotic events in combination with thrombocytopenia were observed in patients who received the AstraZeneca vaccine. And the thrombotic complications were typically cerebral vein thrombosis or splanchnic vein thrombosis in the 2nd to 3rd week after vaccination. Soon after, 3 reports had been published in the New England Journal, one from Scandinavia, from Norway, one from the UK, and one from Germany and Austria. Those of you who are interested in the details and the largest case series are referred to this paper in the New England Journal from August this year, where Sue Payford summarized more than 250 patients in the UK, the largest patient group reported up to now. The same issue or the same problem after vaccination had been reported after the Johnson & Johnson vaccine, also an adenovector-based vaccine in the US, although it seems that the frequency of the complication after this vaccine is somewhat less frequent than with the AstraZeneca vaccine. I have already mentioned at the introduction that all vaccines can induce ITP. How to differentiate ITP from VITT? The typical pattern there is that ITP has a low platelet count, and usually the D-dimer levels are normal or moderately elevated. All vaccines can induce normal thrombosis. Here, the platelet count is typically normal, and D-dimer is moderately elevated, although in case of severe pulmonary embolism, platelet counts can decrease as well. And a typical constellation for VITT is that the platelet count is decreased and the D-dimer levels are strongly elevated, usually more than 4 milligrams per liter. The hallmark of VITT are anti-PF4 antibodies. And it was great luck for the medical community that the available assays to diagnose heparin-induced thrombocytopenia allowed cross-reactivity testing of VITT antibodies. High heparin concentrations inhibit the reaction, but other than in heparin-induced thrombocytopenia, the VITT antibodies also bind to PF4 alone. Relatively soon after the first descriptions, it became clear that not all HIT assays react the same. They have different sensitivity for these antibodies, and generally spoken, the rapid tests, especially assays in which the antigen is coated to beads, do not work and give false negative results. This was confirmed by the French group and also by a German consortium. So in summary, the PF4 polyanion ELISAs work, but they have different sensitivities, and rapid tests, especially if antigens are coated to beads, produce false negative results. The explanation for this has elegantly provided by the group of Donny Arnold, Isaac Nazi, and John Kelton in Hamilton. They have shown that VITT antibodies bind to the red patch on PF4, which is the same binding site as heparin binds to PF4, while the HIT antibodies bind to the bluish spot and epitopes here. And most likely, the way how PF4 is immobilized on the solid phase has a major impact on the sensitivity of these assays. Functional assays are highly relevant in VITT, like they are in heparin-induced thrombocytopenia. These were the index patients we reported right at the beginning, and you can already see that if some sera activate platelets even in the absence of heparin or anything, and very important and much in confirmation of the findings of the Hamilton group, heparin is inhibiting the reaction because it competes with the antibodies for the same binding site. PF4 is strongly enhancing the reaction, and important, immunoglobulins, IVIG, is inhibiting the reaction. And these initial observations here in the right side, in a somewhat larger group of patients, led rapidly to the recommendation to use IVIG as one of the mainstays of treatment in these patients. This has been reproduced and confirmed by the French group and also by the group in Hamilton. And I selected this slide because the French group also added another experiment by which they removed a glycosylation pattern from the immunoglobulins, from IgG, and this fully inhibited the reaction. And this proves now that the activation of platelets is really induced by antibodies and activating platelets via the FC receptor. In summary, washed platelet assays plus PF4 work. Washed platelet assays plus heparin are insensitive and give false negative results. So the VITT pattern in the literature is thrombocytopenia and thrombosis 4 to 20 days after vaccination with an adenoviral vector-based SARS-CoV-2 vaccine, strongly positive PF4 heparin IgG ELISA, and it is Fc gamma receptor mediated platelet activation enhanced by the presence of PF4. Now I come to the unexpected. And this 40-year-old previously healthy female patient. She presented at day 7 after vaccination with 2 days of chills and severe headache. Her platelet counts were moderately low. The dimer was not tested. CVST was ruled out by CT scan, and she was discharged by a painkiller. And this was not a good idea because 3 days later she was readmitted with a Glasgow Coma score of 12, thrombosis in the right jugular vein, the pulmonary embolism, and secondary cerebral bleeding. Her platelet count now was much lower. The dimers strongly elevated. And we identified a whole series of patients who all showed the same pattern. They presented with severe headache, thrombocytopenia, but no overt thrombosis. All patients who were sent home with symptomatic treatment of pain, of the headache, developed severe thrombosis, mostly CVST, during the next days. Those who had then been immediately treated by IVIG and anticoagulation did not develop further thrombosis. And this means we have a 2-time period pattern of headache in VITT. Very frequent, and one of the most frequent adverse reactions after vaccination is headache at the day of vaccination or the following day. They are harmless. A warning sign and a red flag for the neurologist and the emergency doctor is delayed onset severe and persisting headache. These patients require anticoagulation and IVIG treatment to avoid progression to CVST. And this also indicates that the headache is probably not caused by the thrombosis. It is either caused by microthrombosis or by inflammation of the vascular bed. The second surprise was the transience of the anti-PF4 antibodies. Linda Schönborn from our group followed an entire cohort of these patients, and within 12 to 16 weeks, the functionally relevant antibodies become negative in the functional test, while the ELISA is positive for a much longer period of time. So ELISA is not appropriate for screening for biologically antibodies. The gold standard in this question are the functional tests. These findings have major implications. The first is the second dose of the AstraZeneca vaccine should not be given before 3 months to avoid situations where someone is getting the vaccine while still asymptomatic platelet activating antibodies may circulate. And this accounts for all patients receiving the AstraZeneca vaccine. The second implication is that these patients can receive a second vaccine shot. We only have experience with the mRNA vaccine, and none of these patients who had full-blown HIT after the first AstraZeneca dose developed further complications when receiving an mRNA vaccine. So in summary, VITT is caused by anti-PR4 antibodies. Microtiter plate assays can detect these antibodies. PR4 binds. The PR4 binding site of VITT antibodies is the same as the heparin binding site. Functional assays confirm PR4-dependent platelet activation. VITT can occur as thrombocytopenia only, and severe headache is a red flag and warning sign indicating a medical emergency. And anti-PR4 antibodies are transient, which has implications for further vaccination. VITT parallels in many regards autoimmune HIT, and those of you are still struggling with this rare disorder are referred to this review article, which still summarizes the most important aspects. You all know from HIT that most antibodies are irrelevant. This is the iceberg model proposed by Ted Walkington. Group 2 antibodies activate platelets in the presence of heparin, and since a few years we know about autoimmune HIT. These patients have autoantibodies activating platelets even in the absence of heparin. They are no longer the tip of the iceberg, they are the flag on the tip of the iceberg. On all antibodies we observe in VITT are reflecting the reactivity pattern of autoimmune HIT antibodies, cross-linking PR4, forming immune complexes, as has also been nicely experimentally shown by the Hamilton group, and then causing the prothrombotic disorder. HIT is a weird immune response. The B cells producing these antibodies require additional antibodies, and in a prospective study performed several years in Greifswald, in trauma surgery and orthopedic surgery, the immune response to PR4 was nearly exclusively related to patients undergoing major surgery, while those undergoing minor surgery, although receiving the same type of heparin, being on the same board, had much less pronounced immune response. This means B cells producing HIT antibodies require co-signaling or a danger signal. Even if we challenge PR4 knockout mice who have never seen PR4 by polymicrobial sepsis, they produce beautiful PR4 heparin antibodies, and when we obtain cord blood B cells from newborns and stimulate the B cells ex vivo, they produce beautiful anti-PR4 and PR4 heparin antibodies. This means the anti-PR4 polyamine antibody response is part of the natural antibody repertoire, and this may explain some of the unusual biological behaviors of these B cells. Now I come to the molecular mechanisms of VITT. Currently, we assume that VITT is caused by a two-step mechanism. The first step is antigen formation and the immunological danger signal, which together activate B cells. Then, after 1 to 2 weeks, highly pathogenic antibodies are formed, which then result in a prothrombotic state and the full-blown picture of VITT. I would like you to follow me to day 1 and 2, antigen formation and inflammatory signal. First, we assess the vaccine. by a technique measuring small particles called dynamic light scattering, and we found 90 nanometer particles. That's the size of the adenovirus. When we added PF4, the adenovirus became larger, indicating that PF4 binds to the virus particles. When we then added, here in bluish, a monoclonal anti-PF4 antibody, these complexes became even larger, indicating that PF4 binds to the complexes. And when we added anti-PF4 antibodies purified from VITT patients, they showed the same size increase, indicating that their antibodies bound. The third group here shows a very important experiment. When we added DNA in low concentrations, the complexes became much larger, and this has major implications for the pathogenesis, and everything is charge related as high heparin concentrations disrupt the complexes. By super resolution microscopy, we can visualize these complexes in magenta, particles stained by a monoclonal antibody against the virus hexon protein, and in green, a PF4 stained, and you can appreciate that both form complexes. We can even visualize the multimolecular complexes of PF4 in green and adenovirus hexon protein particles, and in bluish here, the affinity purified anti-PF4 antibodies of VITT sera. This is visualization of the complexes which cause the prothrombotic disorder VITT. When we look at the vaccine, we found the chimpanzee virus particle, but a lot of amorphous material around. And when we added PF4, the amorphous material clustered, forming complexes. And these complexes stain by gold particle monoclonal antibodies for the hexon protein of the adenovirus, and on the right-hand side for PF4. So we have large complexes exposing hexon protein and PF4 together, and they are probably the substrate which is inducing the immune response. But where comes the co-inflammatory signal from? The AstraZeneca vaccine is not clean. It contains a lot of proteins. The bluish ones here, as analyzed by proteomics, are the chimpanzee virus proteins. The gray and green ones are the proteins derived from the hex cell in which the virus is propagated, and even a few spike proteins were found, which means that the vector is a bit leaking in the cell culture. Half of the proteins in the vaccine are in fact from the hex cell line in which the virus is propagated. But in addition, when one calculates the amounts of proteins, there must also be quite a large proportion of non-assembled hexon proteins in the vaccine, and together, the hex cell proteins and the free virus proteins, in our opinion, activate the innate immune system, causing the inflammatory response. How do these particles get into the vasculature after intramuscular injection? When we inject the vaccine induces intradermally in mice capillary leakage. The same happens when we inject EDTA, and the vaccine contains 0.1 millimolar EDTA. In fact, within half an hour after vaccination, we find the virus all over in the mouse. And some individuals vaccinated react rapidly with these skin reactions and capillary leakage causing swelling, which remember serum sickness disease. When we now, 1 or 2 weeks later, isolate the antibodies from these patients, these antibodies do not activate platelets alone, they activate platelets in the presence of PF4. But when we added DNA, they also activated platelets. The question is now, where is DNA coming from in the circulation? The answer is it is coming from neutrophils, which release nets, undergo netosis, and thereby release DNA. When neutrophils are incubated with platelets in serum of VITT patients or the purified VITT antibodies, they activate strongly neutrophils which release DNA, PF4 binds to the DNA. The anti-PF4 antibodies bind, forming immune complexes which further activate platelets and neutrophils and monocytes, which altogether result in a strong pro-inflammatory disorder and pro-aggregatory disorder resulting in thrombin generation. And that netosis is also true in vivo, is shown by elevated markers for netosis in VITT patients like cell-free DNA, myeloperoxidase, or other markers. We had the chance to investigate cerebral vein thrombi, either obtained by thrombectomy or after autopsy. And when we stain these thrombi, they are full of neutrophils and nets, mimicking in vivo what we have observed by the in vitro experiments. I'm now coming in the final minutes to the unknown. The first is that we also observe ITP, and we currently address this question by asking whether platelets are the antigen-presenting cells in VITT and ITP. We can measure binding of the vaccine or vaccine constituents to platelets, and probably these platelets then transport the antigen to the marginal zone of the spleen, where B cells get into contact, and we see co-presentation of virus epitopes together with platelet epitopes, and thereby causing misproduction of platelet autoantibodies. A second hypothesis we currently follow is what is happening with all those proteins which are in the vaccine. And the green proteins here are membrane proteins of the HEC cells, not all extracellular outer cell membrane proteins, but also intracellular membrane proteins. And the HEC cell comes from one individual, and proteins are polymorphic. And we know from the veterinarian area that when a vaccine is derived from a cell culture of the same species, that contaminating proteins of the cell culture cells can induce immunization in the animals, later causing severe fetal-maternal incompatibility by immunizing the cows against a platelet epitope of the calf. We have no idea whether this happens with the AstraZeneca vaccine, but these questions need now to be addressed. So in summary and conclusion, it is caused by anti-PF4 antibodies. Human proteins and EDTA in the vaccine likely contribute to trigger the immune response, EDTA by inducing capillary leakage, human proteins and free virus particles by inducing the pro-inflammatory stimulation via innate immune receptors. Anti-PF4 autoantibodies activate platelets, and activated platelets recruit granulocytes. Activated granulocytes undergo netosis, release DNA, and this amplifies the reaction and causes a thrombin burst. What is unclear in the moment is whether co-presentation of vaccine constituents and platelets cause a misdirection of the immune response to produce anti-platelet autoantibodies, and whether vaccine-induced immune responses are also generated against other proteins which may be relevant for fetal-maternal incompatibility. Ladies and gentlemen, I had the privilege now to present you these data, which have been generated by a group of very enthusiastic physicians and scientists in Germany, but also with the contribution of colleagues from other countries, including Canada, where Ted Walkington played a major role, who worked together for weeks to produce rapidly these data to help the medical community to better understand the adverse reactions of VITT and to contribute somewhat to the new treatment approaches. And I'm now happy to take questions.
Thank you so much, Professor Greinacher, for this wonderful presentation, very comprehensive. Obviously, the topic is very complex, and you've dissected really nicely, not only the history, but also the clinical presentation, the typical and atypical, the diagnosis, the variability in testing, and recommendation for how to make an appropriate diagnosis, but also the molecular mechanism behind the syndrome, which is clearly complex and multi-layered. So thank you so much for doing this. Now, we had hoped that Professor Greinacher would be with us to answer the question for a Q&A period. Unfortunately, I just received the word that he's unable to be with us today. However, please post your question in the chat box, and we will collect those questions and communicate with you through Professor Greinacher and give you those answers. So thanks again for listening to this comprehensive talk. I also know that the recording will be available online later on. So thank you so much. And I think you now should stay tuned for the next presentation, which is from our leadership, Dr. Susan Kan and Dr. Mark Roger, presenting as co-director of CanVECTOR. Thank you.